Good evening, Shoals Lab community. All right, welcome to the Shoals Marine Laboratories weekly marine science seminar. I'm Carrie Keogh. I'm currently one of the co-instructors for the Research in Biology course. Alongside tonight's speaker, Dr. Rebecca Atkins, and our wonderful TA, Paige Greens. Thank you so much for joining us tonight for this evening's seminar, our 10th Rock Talk of the 2024 season. These seminars are affectionately referred to as Rock Talks, in part because back in the early days of the lab, seminars and classroom lectures would often be conducted near the intertidal on the rocks. Although we now all meet in the commons, these weekly seminars provide a wonderful opportunity for our island community to come together and learn about a range of topics related to the marine environment. For those of you joining us online who may be new to our lab community, SML is the oldest and largest marine lab in the country that is focused on undergraduate training, both in coursework and in research. The lab is operated jointly by the University of New Hampshire and Cornell University. In addition to our guests joining us remotely tonight, we have 3 courses on Appledore right now: Research in Biology, Marine Environmental Science, and the Biology of Sharks, Skates, and Rays. There you all are. We also have our Shoals Undergraduate Research Group, affectionately known as the Surges, who are now in week 9 of their summer-long research experience. We are excited to have everyone, both in person and online, joining for our seminar tonight. The format for tonight's seminar is a 45-minute talk, followed by 15 minutes of Q&A. For our friends joining online, please use the Q&A box at the bottom of your Zoom screen. We will read your online questions out loud to the entire audience. For those of you in the commons, we will pass around a microphone so that everybody can hear your questions. Okay, so this evening's speaker, I have the pleasure to introduce Dr. Rebecca Atkins. When she's not serving as a faculty member for Research in Biology, or simultaneously as one of the Island Scientists in Residence Fellows mentoring the SURGE group, she is formally a scientific program manager with the National Oceanic and Atmospheric Administration, or NOAA. I had the pleasure of getting to know Rebecca when we overlapped at the Odom School of Ecology at the University of Georgia, where Rebecca completed her PhD in 2022. She's going to give us some background on her academic journey as part of her talk. So you'll get to learn a little bit more about her earlier work and her career trajectory in just a minute. Rebecca has published her work in both her undergraduate and her graduate work in the Biological Bulletin, Oikos, and the Marine Ecology Progress Series. After completing her PhD at UGA, she was selected as a Knauss Marine Policy Fellow. And as is typical for Rebecca, she shot right to the top, and she worked directly with NOAA's Senior Advisor for Coastal Inundation and Resilience to help facilitate the strategic planning and coordination of all coastal resilience activities across all of NOAA. At the conclusion of her Knauss Fellowship, Rebecca transitioned to her current role, where she serves as a scientific program manager for the National Center for NOAA's National Centers for Coastal Ocean Science, particularly for the Competitive Research Program. Here, Rebecca is putting her considerable organization, strategic thinking, can-do attitude, coalition building, and communication skills to use to help NOAA coordinate their competitive research programs and allocate funding for coastal resilience research. Her work includes a range of program management activities across the research programs to achieve agency and stakeholder needs, including development of short and long-term research program plans, oversight and control of efforts to support each project's requirements, procurement, development, testing, implementation, publication, and briefing of relevant project results to leadership, external partners, and congressional audiences. We'll get to learn a little bit more about her work at NOAA at the end of her talk. For the last 2 years, Rebecca has been one of the scientists in residence serving on island to mentor the Surges. In this role, she helps these amazing undergraduate researchers develop new tools for their portfolios, develop strong and relevant research questions, troubleshoot problems, analyze data, and prepare to communicate their work. She also serves for them as a field technician sometimes and as a boat captain to facilitate their projects. This is the 3rd year she's been at Shoals as a faculty member for Research in Biology, which has been truly a pleasure. She loves to talk to students. She really loves it. And I hope that some or all of you students will take advantage of the opportunity to learn from somebody like Rebecca, who has a great wealth of insights, knowledge, and passion for what she does. So it is my immense pleasure to welcome to the podium my dear friend and colleague, Dr. Rebecca Atkins, who will present her research from snail breathing to coastal resilience, an early career quest. Please give it up for Rebecca Atkins.
Thank you, Carrie. I think that you covered the career stuff, so we probably can just dive into the science and we'll be fine. Oh, thanks, Kaji. And when Carrie said that I helped to mentor the summer undergraduate research group, what she meant is that I learn from them every summer because they're actually the ones that are teaching me more so than the other way. I truly believe that. So with that, we'll just dive right in once I figure out how to. Yeah. General outline, kind of like Carrie mentioned, the first part is going to be a little bit of a brief timeline, how I got here. I'm really excited to be doing this rock talk today so I can share a little bit about what brought me to Shoals in the first place. And this has also been like a pleasure attending these talks throughout the entire summer. We'll then go into a little bit of research rabbit holes because I would be remiss if we didn't talk about science during a rock talk. And then talk a little bit about NOAA, some early career development, and hope that maybe you can see yourselves taking a similar path in the near future. So we'll start with some earlier research experience. I went to the University of Florida, started there in 2010, and was working on a bachelor's of science in wildlife ecology and conservation with a minor in fisheries and aquatic sciences. And this particular program was great in that there was a lot of experiential learning that prepared us for careers in wildlife ecology and conservation biology. And then I also worked at the Office of Sustainability, helping to run some campus-wide recycling and composting efforts. That's also another reason why I love Shoals so much. But when I say experiential learning, we could be doing anything from tagging monarch butterflies to mist netting for birds, to helping with manatee health assessments. There was so much that you could do as an undergraduate to just dive in and help other graduate students do their work, or volunteer or be an intern. So I'll talk a little bit about some of those other experiences as well. But undergraduate and high school was prime time to just try a lot of different things. One of those things for me in undergrad too was a research experience for undergraduate program, also called Talk to. I did that in the summer of my sophomore year, and it took me to Sapelo Island, Georgia, where I experienced my first salt marsh. And it also happened to be co-located with a long-term ecological research program, LTER, the Georgia Coastal Ecosystems LTER, which brought together scientists from all over the country doing really cool work in the salt marshes. Super inspiring, and that's where I did a really cool study in salt marshes and published that work, which helped segue into graduate school. The summer after my REU, decided to go to Wales and look at macro algal diversity in rock pools because I had talked with a postdoc during my REU who was getting a lab started up in Wales and just wanted somebody to come and do a little bit of early research. So good opportunity just to say yes and dive in and go for it. It was super fun. When I graduated, I was pretty sold on research as a path that I wanted to continue to explore. And so I had lined up a summer internship at the Gulf of Maine Research Institute, which now actually runs their own REU program. And there I worked alongside a lot of other undergraduates that were doing really, really cool work, things like studying alewife migrations through rivers, acoustic monitoring, and measuring the caloric value of different fish species. And I got to help set up a survey protocol for quantifying the abundance and diversity of juvenile fish across different habitats in Pasco Bay. And this included pulling that giant seine net that you see there across all these different areas to see and compare juvenile fish hanging out at the edge of habitats like salt marshes, tidal marshes, seagrass beds, pebble beaches, mud flats. It's a really cool way to kind of see what was there and get to help contribute to what is now a long-term monitoring effort that they've engaged with for the last almost 10 years now. Makes me feel old. After working at GMRI, I then decided to reconnect with a professor that I had met at the University of Florida, who had since moved to the University of Georgia, and I wanted to work as a technician in his lab. And so I started by helping him with data management, which is less glamorous, but still really cool and important work, helping him to catalog and document some of his long-term data that he'd collected on coral reefs in French Polynesia. And during that time, he encouraged me to apply for the Graduate Research Fellowship Program, also known as a GRFP, which is something that you can apply for to get funding to go to graduate school. And you can apply for once before you're in graduate school and then once when you're in graduate school. Not entirely sure of the rules, but Kyla's going to be talking about that. I highly encourage you, if you're thinking about it, to sit in. And so I applied with Craig, my mentor's help. And then when I received the fellowship, I also had already reached out to other potential advisors, mentors, to think about what other institutions or labs might be a good fit for my next step. And I decided at the end of it to stay with my current mentor at the University of Georgia and continue to work in the salt marshes that I had grown to love the previous several years. And while I was in graduate school, I also realized that towards the end of it, I was interested in how we apply science to solve real world challenges and make sure that the best science is being used to inform policy. And I also had no idea what life was like outside of academia at that point. And I had spent most of my time really diving into exploring basic curiosity-driven questions about the marsh. So I applied for the Marine Policy Fellowship and spent the last year of graduate school living in Washington, D.C. and supporting NOAA's senior advisor for coastal inundation and resilience, and joined another cohort of amazing graduate students who got placed across different agencies and across legislative offices as well. So I'll touch a little bit on the NOAA work at the end, time permitting, but now it's time to dive into some research rabbit holes. And I want to preface this with acknowledging that so much of the work that we do as curiosity-driven scientists revolves around recognizing patterns and relationships in the environment around us, and then trying to dig into that so what or why. And I spent a lot of time during graduate school trying to kind of finally adjust the crosshairs of my research, so to speak, starting out with really broad observations and then quickly went in a lot of different directions. And so I'll take you down an abbreviated version of some of that work and sprinkle in a little bit of ecological theory along the way. And we'll focus mostly on the importance of thinking about spatial scale when exploring ecological systems, and then also thinking about different levels of organization, like how we get from individuals, populations, to species interactions. And I want to be upfront that the purpose of this is not to try and have you leave with specific information about what I chose to study, but to give you a sense of how you might go about asking some of your own questions and some potential methods that you could use in doing that. So let's zoom into coastal marshes in the southeast and mid-Atlantic. Is anybody here familiar with salt marshes? You've been in one. Okay, sweet. Salt marshes are awesome places. They're beautiful ecosystems. They're not only incredibly productive and help us sequester large amounts of carbon, but they provide valuable ecosystem services, probably like you already know, in the form of recreation, valuable fisheries, habitat, and coastal protection. Tonight, though, we're going to zoom in really closely on a very specific niche aspect of these marshes, and that is the salt marsh periwinkle, Littoraria irrata. And these are one of the most abundant grazers in the marsh. They're present year-round, and they also exist alongside their grazers, like grasshoppers, sucking insects, and herbivorous ferns. And one of the ways in which Littoraria can affect cordgrass is by inflicting these grazing scars using what we call a radula, which is essentially a regenerating ribbon of teeth that they use to rasp away at a substrate. And so if snails were any larger than they currently are, this probably would be the stuff of nightmares. But in the case of cordgrass, it means that they end up creating these pretty visible grazing scars that you can see here. And if you notice, these scars are a little bit brown around the edges. They're starting to decay, and they're actually usually infected with a fungus that is particularly nutritious to these snails. And snails can even facilitate fungal growth when they graze and create scars, and then they poop on those scars. And that contains fungal spores that then gives rise to additional growth of fungus. And so it's kind of like a 1-2 punch to these plants when the snails are grazing and they're leaving the plant not only with a scar, but with a fungal infection. So when I went into the REU experience that I mentioned in my sophomore year, I just happened to land in the lab of somebody who studied these snails with a particular emphasis on their negative top-down consumer effects. Their work highlighted these snails as a dominant consumer of the marsh, and they characterized a phenomenon in which a lot of these littoraria can quickly graze down a large area of Spartina until there's nothing left but a bare patch of mud that can take seasons to recover. And these are really interesting results that we dug into. And it shows that these grazers, even something as small as a snail, have some pretty huge effects on the environment. And just to note, I'm going to interchangeably use consumer resource interaction and consumer effect and sometimes top-down control to describe essentially the relationship between snails and Spartina. And I'll also often refer to consumer effects on plant biomass specifically, with negative consumer effects occurring when snails are suppressing plant biomass. So you'll see that below the zero. And then also thinking about positive effects when they facilitate plant growth above what would be otherwise naturally occurring if the snails were not present. So positive, facilitative, or strongly negative. And when I went into the field, I was really cognizant of the work that my graduate advisor had spent time working on. However, in addition to that work, the strong top-down control work, I was also really cognizant of the fact that everywhere I walked in the marsh, I was seeing snails. They were on the mud, they were on the stalks. And they also serve as really valuable parts of the food web as food sources for crabs, fish, birds, turtles. They also play a role not only as grazers, but they consume standing dead plant material and they help recycle nutrients in the marsh. And when I looked through previous studies, folks other than my mentor at the time, I noted that there was a lot of work that had found that the effects of these snails sometimes are neutral. Sometimes they're positive and they're affecting things like plant productivity and reproductive growth. And so clearly a lot of nuance in how these snails are living in and influencing their environment. And there's a lot of marsh where these two species are co-occurring. And so at the beginning of my time as a graduate student, I wanted to figure out how generalizable this top-down consumer control paradigm in this system was, given that we have studies that have been done in different places at different times, but all find different things. And so I spent a lot of time in the first year of grad school scouting out different sites, going to different marshes along the coast and seeing where I could access and work in larger areas of the marsh. And I wanted to capture as much of Littoraria's geographic range as possible so I could really characterize what the role of Litteraria was in the system as a whole and what they're doing in the marsh. And this happened to take me along approximately 900 miles of coastline, which was awesome. I went from working in a very small island off the coast of Georgia to trying to think about expanding this out to the range of the species. And as you can imagine, there are a lot of things that might vary at the spatial scale, things that we might need to consider. I'm just gonna like poll the audience if y'all can think of anything to vary along that kind of a spatial scale. Yeah. So good. Temperature. Yeah, what else? Totally. Salinity, humidity, native creatures, species. Yeah, species turnover. No, totally. I completely agree. Anything else? These are all great. Weather, season. Yeah. Sure, some areas might be affected by storms. This is just kind of getting our brains jogged and thinking about, when you're working along these really large spatial extents, you also potentially are encompassing a lot of different things that might be changing. And it's something to keep in mind. So in addition to thinking about the effects of the snails on the marsh, I was also trying to think about other things that might influence the relationship that we're seeing. And this perspective could be described as macroecology, which is generally the study of broad scale ecological patterns and processes. And so when we're looking at consumer plant interactions through this macroecological lens, empirical studies generally assert that as we move towards these lower latitudes or towards the equator, consumer pressure becomes more intense. And this is a pattern that can be linked sometimes to biodiversity and differences in the number of species that are there. And it can also be linked to climatic conditions like temperature, humidity, salinity. And in this case, I was particularly interested in a temperature angle. So a recent meta-analysis, which is just a study of studies, it's an analysis of previous work that has been done, has also suggested, this came out in 2016, right around the time I was starting graduate school, and it suggested that even marshes, within marshes, consumer effects are likely to be strongest at southern sites, and that this was true even for things like snails and crabs and insects and other ectotherms that are responsive to their environment and the temperature that they're in. And so this study attributed that effect to changes in temperature as you went from northern latitudes to southern latitudes. And so I expected that I would find that as well in Literaria. And I wanted to note, we're going to be revisiting this figure several times. So consumer effect is on the x-axis. There's a 0 at the top, which indicates neutral effect. Anything above that would be positive. Anything below that would be negative. I came in with the assumption that we'd probably be seeing negative effects based on my previous mentor's work, and that there would be this linear relationship where when you go towards the pole or northern latitudes, you'd see a less negative effect. So going towards 0, and then latitude is on that x-axis. So now's the fun part where we can talk about experimental design, which is something that we desperately dug into in research and biology. And I'm assuming other classes are probably going to be touching on that soon, if not already. And I selected 8 sites to work at in the geographic, kind of spanning the geographic range of Literaria. And I wanted to assess the effects of snails in marshes at their ambient or naturally occurring densities, not anything that's artificially stocked, but what's just there naturally. And so I needed a way to compare how what kind of biomass does when snails are present. And to do this, I set up cages where I excluded the snails from the cages, periodically going back and removing them. And then to account for the effect of having a cage in the marsh, I put out what I called a cage control. It was a 2-sided cage that allowed for snails to move in and out, and it mimicked other effects that the cage might be having in the marsh. So I could compare a plot that had natural snail densities with caging material to a plot that had caging material but no snails. And so that's the comparison you're going to see. It'll be of Spartina above ground biomass in snail removal plots compared with the 2-sided cage control. And to make sure I was also accounting for other things that could have given rise to variability or that might vary as a function of latitude, I also measured things like pore water chemistry, which can influence plant health and productivity, crab and mussel abundance, which can also influence plant health because things like mussels and crab burrowing can oxygenate the soil, help with nutrient cycling, and promote plant growth. And then also measured Literaria population characteristics like their body size and abundance. And so here you're going to see a plot of above ground biomass that's going to be on the y-axis and measured in, you'll see a measurement of above ground biomass in the removal cage, which is going to be shown as a gray circle versus the 2-sided cage, which is 2-sided control plot with snails present in green triangles. And so there's going to be lines connecting them so that you can better visualize how biomass changed between those 2 treatments. And on average, snail removal had no effect. If you just took the average across all of those sites, when you remove snails, there's no change in above ground Spartina biomass, which is not what I expected to see. However, even though on average there was no consumer effect that we detected, there was still differences across sites in both the direction, so positive or negative, and the magnitude of the effect of snail removal. So some sites, you did actually see biomass improve when snails were present, so a facilitative effect. Others, we saw it decrease, the ones that I kind of show here in orange and red. And my original question goes back to, how does this look when you plot it along a latitudinal gradient? And this was my hypothesis that we started out with. Consumer effects would be more strongly negative at southern latitudes. But what we actually found was a significant unimodal relationship. So at the ends of their geographic range, particularly in the southern end here where you see close to the 0, they have neutral or positive effects potentially. And then when you get towards those middle latitude sites, you see stronger negative effects of Literaria. And at that site that's at the very, I don't know if you can see that, yeah, very bottom there, plant biomass was actually reduced by about 60% when snails were present. And so a huge take home here is that if you just were to average across the effective Literaria at every site, you wouldn't see a consumer effect, or you would assume that there's no consumer effect. Also, depending on which site you chose to work at, you might get totally different answers when it comes to the effects that snails are having in these marshes. But by considering multiple sites and the potential for some large-scale patterns, we open the door to additional questions, such as what's giving rise to this unimodal relationship? One reason could be that consumer populations also vary across site. And in particular, I'm going to walk you through thinking about metabolic biomass. biomass, which is just a fancy way to think about the energetic demand of that entire population of snails that was in a plot, or how much energy is required by that group of snails to survive, grow, and reproduce, would translate to how much food they need to take in. And we often calculate metabolic biomass by summing across the mass of every individual in that plot, which is raised to the 3/4 power. We're not going to dig into it, but essentially that's the transformation that you would apply in order to account for the fact that smaller individuals have a higher energetic demand per gram of body weight than larger individuals. So that's what that 3/4 power scaling is doing. They're applying that to biomass. So first, in order to think about population metabolic biomass and how it might differ, we need to go back and look at our snail population traits to see if we see any latitudinal patterns there. So on the top, you're going to see population density that was recorded in these different plots that I was working in, and then shell width on the bottom plot, latitude across the x-axis. And up top, you can see a unimodal relationship with high snail densities at mid-latitudes. And then a really tall point right there is actually Sapelo Island, Georgia, with the highest snail density recorded. And then you can also see that average shell width increases with latitude, which means that on average, snails are larger at northern sites. And this is a pattern that has been observed across a lot of different ectotherms. And it's known as the temperature size rule, or it's commonly referred to as the temperature size rule. And then when we convert that, so we take how many individuals are there and what their masses are, and we convert that into our metric of metabolic biomass at that plot level, we see a uniform relationship. So peak metabolic demand of the snails that are in these plots at these mid-latitude sites. And so this led me to wonder if snail metabolic biomass changing across latitude could give rise to that pattern that we see in consumer effects. And it's worth noting that the RAU project that I did on Sapelo Island, I did find a significant negative relationship between metabolic or consumer population level metabolic demand and consumer effects. That was something that I also looked at as an undergraduate. And it stands to reason that we might see this across sites, not just when we're looking within a site. And to some extent we did. You can see this negative relationship between consumer effect and metabolic biomass. And it conforms to what we previously found or knew about the system. However, it's not statistically significant. Doesn't mean it's not biologically significant, but it's not statistically significant, which also the variability that you see here, that's not forming a perfect line. There's variability around this relationship, which makes you think that there's a lot of things left to be explained. Otherwise, like I said, everything would be a perfect line and we couldn't explain anything else in there. So based on this, we know that it's not just metabolic biomass that's driving the relationship between snails and Spartina across latitude. Somewhat maybe, but it's not the whole story. So we could ask ourselves a lot of different questions here, going a million different directions. What might be driving variation in snail metabolic biomass? But also take another step back and ask, how are we even calculating metabolic biomass in the first place? I explained a scaling exponent, but that also just assumes that everything's constant across all the snails and all the populations. And so we might need to think a little bit more about how sites might vary from one another in terms of metabolic traits of individuals. And that can inform how we scale up individuals to populations. So I'm going to segue a bit into thinking about how we estimate energetic demand and if that might vary across individuals and populations and different sites. And so we saw previously natural snail populations in the field. They're made up of individuals of a lot of different sizes. And I wanted to better understand how feeding rate might vary as a function of body size. And snails in the field, like we also vary in the temperatures that they experience. Sites are very different. And so that can also influence metabolism and metabolic demand. And I also need to note that this work would not have been possible without two amazing undergraduate assistants, Will Ellis and Kat Clancy, who helped in the field and in the lab. And they were co-authors on the paper that got published with this work. And so often when we study temperature and metric of performance like feeding rate, we refer to this or refer to it as a thermal performance curve, which is an expected unimodal relationship that demonstrates how organisms have an optimal temperature that we perform at. And then if you go away from that optimum temperature, performance will decline. And we can build body size into this by thinking about how larger individuals, here represented by a blue curve, have a higher overall performance or feeding rate. And then smaller individuals represented by the black curve have a smaller feeding rate or performance. And the distance between those curves, how much a snail or how much performance varies between small, medium, and large individuals is referred to as size scaling. In this case, how much it's changing with size. And both the size scaling and thermal traits that we can derive from experiments that characterize these curves can go into an equation that allows us to scale up individuals to population level performance when you take into account temperature as well. And in this work, I'll talk about feeding as the metric of interest. So a little bit of the fun stuff too. Like I said, Kat and Will were awesome at this. To measure snail feeding rate and relate it back to body size in the first place and temperature, we had to figure out how to quantify snail feeding. I told you that they create these grazing scars, but they also can consume in a lot of different ways that might not be as obvious as a grazing scar. So we needed to figure out how to standardize the substrate that they were feeding on so that we could quantify feeding. And so that required making snail gelatin, which basically consisted of dried pulverized spartina that we added to agar, creating this delicious gelatin that was then poured into Petri dishes. And then those Petri dishes, we had placed a snail of a certain size into it, and then went into incubators that were set at different temperatures. And we let the snails do their thing for about 72 hours. And after that, we measured the jello consumed using a program called ImageJ. Totally great, check it out. And then we calculated feeding rate as area consumed over the 72-hour period. And I say we, Will and Kat did a lot of this work. And so going back to this figure, as a reminder, these are the thermal performance curves. Blue is large, black is a small individual. And I'm going to show you the thermal performance curve for feeding across these three different body sizes. First for Florida. So you can see how feeding rate increases with body size. That's what we would expect. And then looking across South Carolina and Virginia, adding those in, you can see how the relationship between body size and feeding, that mass scaling, the differences between those colored curves is different across these populations, which is something new. We didn't actually know this for this species of snail. And so using this, we can see that the application of a single thermal performance equation, which assumes constant mass scaling and thermal traits across populations of snails spanning 900 miles, would likely lead to a miscalculation of population level metabolic demand. So this exists as a potential source of variation that could help us to refine our comparison of population metabolic biomass across sites and perhaps help to better explain differences in the consumer effects that we measured. Another place that I was interested in exploring, another piece, was predation. And so we know that predators can change how individuals behave and forage. So if there's a difference in predation pressure across sites, we might also see deviations from what we would expect based on population metabolic biomass alone. And here you see pictures of a few of the common predators you would encounter in a salt marsh, super vicious. We have the diamondback terrapin, it's in the top, a blue crab that's on the right, a crab below that. And then to the left, you'll see a picture of a bunch of snail shells that are actually snails that have been plucked out of their shells, likely by a mud crab, and just deposited around its burrow. So indicating super hungry crabs in the marsh. So it's a dangerous world for a snail out in the salt marsh. And I'm not going to dig too much into the weeds here, other than to say that we assess predation pressure across and within sites to get a baseline sense of how it varied. Again, there's not a ton of work that's been looking into this particular species and consumer resource or crab snail interactions. And so we wanted to do this and get a nice snapshot across 4 different sites. And so we tethered them. This was a snail tethering experiment. So we basically tethered snails to sod stakes after measuring their body size. And we put them out across the marsh, across an elevational gradient in the marsh. And we tethered approximately 3,400 snails for this. And we super glued a lot of fingers together. And it would not have been possible without a colleague pursuing his master's degree and another undergraduate field assistant who was also interested in working in marshes and thinking about predator pressure. And so the data you're about to see has latitude on the x-axis and proportion of snails consumed on average along the y-axis, and the points are colored by elevational zone in the marsh. And here you can clearly see that predation pressure differs across sites. The highest predation is happening in the South Carolina site, the top, followed by Virginia, Maryland, and Florida. And this just starts to open the door for us to think about how predation varies across sites and how that different predation pressure could influence things like behavior and foraging like we talked about. And maybe could be influencing snail-plant interactions. And it almost looks like we could maybe detect some sort of a pattern, possibly a similar pattern to what we noticed with latitude. So this is worth digging into a little bit more. If anybody wants to go out and tether some snails, let me know. And you'll also notice differences across elevation, which suggests there could be some within site variation in how snails are behaving or distributing themselves across the marsh. And marshes are generally very expansive places in the southeast and mid-Atlantic. There can be a lot of variability in above ground plant biomass and how snails are distributed that could also give rise to expected differences in consumer effects at this scale, the scale of within a site. And so for the last piece of this, we zoomed back in to think more about scale of a snail. So taking a second to put on our snail goggles, what do y'all think might matter to a snail? What's important? Food. Okay, yeah. Constantly looking around for food. You know, totally. Yeah. Whether or not they're attracted to other snails, want to be around other snails. There's some work that's been looking at how they follow each other's slime trails. It's kind of cool. Yeah. Predator avoidance. Absolutely. Where they lay eggs. And these are kind of cool, actually. They end up releasing veliger larvae into the water column, and then they float around for like, I can't remember, 4 weeks or so, float back in and settle. But yeah, they have a pretty unique pattern or behavior when they decide that they're ready to release their larvae and climbing stalks. And to avoid predators, they also will climb stalks, particularly as the tide is rising and falling. So thanks, snail goggles. Just thinking there are a lot of things that matter to these snails that could influence how they move around in a marsh, where they're at any given time. And so I was asking, does snail distribution vary across smaller spatial scales? And you're right, a lot of different things could matter, and the scale of those different factors could influence the variation that we're seeing within and across the marsh. And to start to get at this, we picked 5 sites, not completely encompassing the latitudinal gradient, but like we did before, but still working along a good range. And this work was also made possible by a high school student, Sam Schaefer, who came out with us in the field to collect this data, in addition to another fellow PhD student in my lab. And within each site, we selected 5 25-meter square plots by randomly selecting latitude and longitude combinations, knowing kind of roughly where we could access in the marsh using Google Maps. And then within that, we sampled 8 1-meter square plots, 6 smaller subquadrats within that. And at that subquadrat scale, we quantified the total number of live and dead Spartina stalks, crab burrows, which is important at predicting their distributions, and literaria number per stalk. So literaria counts were collected using 2 different approaches. One, the number of snails per stalk, and the other one, we subsampled the total snail biomass in 262 of those plots. So I'm not going to dig into the stats today, but I'll show you the raw data, show you a little bit about what we found. So on the x-axis is latitude, y-axis. On the left plot is snail biomass per subplot, and the right one is average snails per stalk. And so the different symbols are different 25-meter square quadrats or plots. And even when accounting for some environmental factors like stalk density and crab burrow density, we still saw a ton of variation among the 25-meter square plots within a site, more so sometimes than the differences across sites, which highlights that literaria distribution may vary more across this spatial scale within a site than among sites, particularly at a couple of places like North Carolina, two that you see here that have a lot of variation within site, which is really interesting to note when it comes to how we measure and understand literaria in typical field surveys and experiments that often are located in smaller areas within the marsh. Maybe they're not capturing the full amount of variability that's out there. And another thing we can do is try and translate this, the biomass that we measured back into that consumer effect metric to kind of see if we were to go out and subsample literaria populations across these 25-meter square quadrats and across sites, how might that compare in terms of consumer effects? And so here you get a sense of how we might expect consumer effects to vary across these spatial scales based on underlying variation and snail metabolic biomass. And you can see again that there's differences that we would expect to see across sites, but also a lot of within site variability represented by these bars of the same color. And so thinking about the implications for how we design our studies and make comparisons, if you were to just work, for example, and a small area within the marsh, and only at a couple of sites, you might conclude here that consumer effects are negatively correlated with latitude. Similarly, if you pick just these two locations, you might see no difference at all across sites. Or if you pick these two, you might assume that there's the opposite pattern with latitude. But you can see here that these particular plots that we pick on the right-hand side are not necessarily maybe representative of the entire site, but you wouldn't know that unless you had made the effort to survey and characterize kind of what that site looked like as a whole. And so the latitudinal gradient represented here, I do want to remind you, it doesn't encompass the gradient that we looked at in the first study. So going back to the original study we started with, it was really important that we worked along the entire latitudinal range and captured numerous sites in between in order to detect that unimodal relationship. Otherwise, that nonlinear effect of latitude would have been completely obscured. And when you dig into it more statistically, in this, there's no single site that's driving this relationship. And so this gives me additional confidence that what we're characterizing is representative biologically of this species and the snail plant interactions in the marsh, even if we didn't fully characterize the range of biomass that could exist within a single site. And it also illustrates that every single site or study or site is a little brick in the building. We can learn a lot of different things from different studies that could apply to what we're seeing at different spatial scales, but it's important to keep thinking about the research that we're doing and the bigger context of the broader system. And so finally, just to drill this in, when it comes down to the latitudinal pattern and litter area consumer effects, there's really no silver bullet explanation to this pattern, but it's probably linked to a lot of different factors. And the same can be for variation happening within sites. And knowing how things work at these different scales can also inform our understanding of how these snails and plants persist, continue to coexist in the marsh, and how they might vary over space and time. And essentially, there's a lot left to explore. That's kind of what I'm saying. So get out there and become a marsh ecologist. And then even as small as a snail can lead to a lot of really cool questions. And it's always worthwhile to let your curiosity guide you. And with my last 2 minutes, I'm just going to kind of take a little bit through the NOAA stuff and some of the early career stuff that I promised. So it's not a total bait and switch. So I'm situated at NOAA, work at INCOS, National Centers for Coastal Ocean Science, as a scientific program manager. And I just want to give you a sense of the sheer variety of stuff that INCOS is doing so that maybe you can see yourself working there one day. It's considered to be a focal point for coastal ecosystem science within NOAA's National Ocean Service. And they do a lot of really cool things related to ecosystem science, ecological forecasting, about harmful algal blooms, environmental stressors, PFAS, coastal resilience and adaptation, and social science, full spectrum of things. And it's not just working in DC, they have labs all over the country. You could be based in Beaufort, South Carolina, Charleston, South Carolina, Oxford, Maryland, or in Citizen Bay, Alaska. I happen to be situated in the Maryland office working in the competitive research program. But some other really cool stuff that INCOS does that I particularly like to focus on that kind of still tethers me, so to speak, back to my original graduate work is thinking about things like sea level rise, how we can model the dynamics of coastal change and understand the impacts on our coastal ecosystems and communities. Applied science, how can we restore and protect our degraded coastal ecosystems? This one's looking at thin layer placement of dredged sediment and helping marshes maintain elevation capital, like how they phrase it. And then nature-based solutions, thinking about exploring the ability to use nature-based solutions as a long-term protection against climate change, naturally adapting and responses. Oyster reefs, another word that for this. So I'm in the competitive research program. A lot of the work that I described to you just now is a lot of work that our internal science team does. But the cool stuff that I get to work on in the competitive research program is funding external science teams. And I really like working with the Effects of Sea Level Rise program, ESLR. We have projects that are active across the country. A lot of these are focused on informing or advancing science to understand nature-based solutions and how they're performing, how they're protecting coastal communities. And some of them also involve partnering with other agencies, the Department of Transportation, to co-fund work that's bringing together really interdisciplinary teams. Think about maybe infrastructure resilience at our coast. And so, in case it wasn't already implied, these teams that we're funding are super interdisciplinary. They're physical scientists and engineers. Diane Foster's talk was a really good highlight of that type of work, bringing together together also coastal scientists and ecologists, transportation professionals, and economists and social scientists to do some really unique work and advance our understanding of coastal ecosystems and actions that we can take to protect our coastal ecosystems using a variety of team members. And so the EASLER program also requires a really collaborative approach between scientists and end users. And this happens at the very beginning of a project to ensure that the information that's being produced by the scientists or the research team is actually usable and can inform decisions that actually need to be made on the ground. And so end users could include natural resource managers, private companies, city and county officials, elected officials, other federal agencies. It's really cool to be a part of this really interdisciplinary collaborative team. And as a program manager, NOAA engages in cooperative agreements. So we work really closely hand in hand throughout the entire duration of a project, to starting from maybe going to site visits and getting to see where they're working, promoting their work at conferences, engaging in their management team meetings. And some of the questions that they get to answer throughout doing this work is, what if we move infrastructure back? Should we move infrastructure back? What are the cost benefit, pros and cons of making these different coastal infrastructure decisions? Where and when should we acquire land? Where are we losing land? Where are we likely to gain things like marsh? Where's marsh migration happening? What if we nourish a beach? What's that going to mean for long-term resilience of that beach or nearby adjacent communities that are accessing it or being affected by it? And so this is like, I'm giving you a really quick and dirty overview of this work, but the variety of approaches that this team supports allows us to really look at what we do on the coast in a holistic and a long-term way. And by having so many project sites around the country, we can also think about the bigger picture of coastal resilience efforts and the science that's needed to inform the implementation of coastal resilience projects, which is particularly important. Almost done, I promise. Last week, or actually this week, the Office of Coastal Management announced a $575 million funding event. They gave out money across the country to support the Coastal Resilience Regional Challenge, and 75 million of that went to the state of Maine. And some of the work they're doing with that is expanding the availability of technical tools and technical assistance. And there's an emphasis on nature-based solutions and the development of shorefront stewards. And so this is a demand signal for nature-based solutions and protecting our coastlines. But we need to make sure that our investments are going to result in lasting change. And so we need good science to inform what we're doing, which is essentially where I view myself as playing a role now. So final advice, I want to emphasize that even though I left academia, skills I gained as an academic have made me successful in this role that I'm in now, working with other scientists, and coming here to teach has also really been enriching for that reason. There's communication that you learn as a graduate student or an undergrad or a high school student. Critical thinking is crucial. Collaboration and partnership, if you can tell with working, a lot of my projects were collaborative, and then now I work with interdisciplinary teams. And then this is just some final advice that I stole from my IBL. They were really awesome. Who you're working with is as important as what you're working on. So I feel really lucky to have this amazing team of people, some of whom I've highlighted below, including Carrie, and support as I make these different career transitions, and talk to as many people as you can, myself included, because there are jobs that you don't know exist, but that you can also create yourself. Like, be your own ecosystem engineer is a fun way to phrase it. And also coffee chats are a good way to talk to people about what they do without, maybe trying to be nervous. Everybody wants to talk about what they do over a cup of coffee or tea. And then here's some additional resources if you want. Previous faculty earlier this year, you can scan the QR code. It takes you to a page full of resources about professional societies to join, job boards. Yeah, literally anything you would probably want to know about grad school and finding a job. But NOAA also has a really cool student opportunity database you can reference based on the audience. If you're high school, if you're undergraduate, if you're a recent graduate, different types of opportunities, and then even down to citizenship eligibility. You can apply if you're a citizen or a non-citizen. So totally check that out. Thank you.
Thank you so much, Rebecca, for such a fabulous talk, celebrating variation in the ecological world, and also highlighting and charting a course for people to figure out how to follow your footsteps. We've got time for questions. And I'll bring the microphone around. I'll also remind folks who are joining online that you can please submit questions in the Q&A box and we'll read them from the computer.
You can also totally find me at any time in the next 2 weeks. Happy to keep chatting.
What was the name of your research paper that you submitted?
The name of the one,
The snails one, for the relationship with the grass,
the kind of thermal performance
Yeah.
curve line. I have to get back to you on the exact name of that.
Okay.
Can't remember it off the top of my head, but I will 100% share it.
What made you decide that like snails were what you wanted to focus on? What got you interested in like the marsh and like that?
So it was a little bit of maybe some momentum that carried me kind of saying yes and, just doing the next logical thing that made sense. And so doing that research experience for undergraduate program put me on Sapelo Island. And then they said, go out into the marsh and see what is interesting to you. And because my mentor at the time had really focused on these top-down snail plant interactions, and I was looking at snails for miles, like, okay, maybe I can like drill a little bit deeper into this and start exploring this particular ecological interaction. But it could have just as easily been anything else. I've known people that have worked on mussels and the facilitation effect that they have on the grass there, people that look at more of the human dimension side of things. And now I'm more into the marsh restoration end of it. But yeah, it just kind of happened. I think that also is a testament to the fact that you don't like let your curiosity drive you. But what you start out, if you start to study something, maybe you love it, maybe you realize that you don't love it, and that's equally important.
I have more of a career-based question. What was your major again? Your career bachelor's?
Wildlife ecology and conservation, and then a minor in fisheries.
I was just wondering, did you ever feel like pigeonholed by your major? Do you think it was worth it to have such a specific major?
Interesting. So I'll have to admit something. I initially was thinking, do like ecosystem or what was it, environmental science. And then I looked and was like, oh, there's a ton of chemistry and a ton of physics, and I'm not feeling super jazzed about that right now. But I also loved being outside and didn't realize that I could potentially have a career working outside, which is what I also kind of thought I wanted to do. And no, I don't think it pigeonholed me because of the fact that there was so much experiential learning involved, and there was a lot of talking to other people and gaining a skill set. Like no matter what, working on different skill sets, even if it's different wildlife survey techniques or working with data or working on communicating about your science, it still gives you a valuable skill set that can be applied in any context. So no, I don't.
So it was more about the experiences of the major.
Yeah, it was kind of a like, it is what you make it.
Thank you.
Also explore a lot. Take so many different classes. I 100% recommend like a women's studies class if you have time.
What's your advice for people who are planning and majoring in like marine biology, marine sciences, that sort of thing?
Ooh, advice. I mean, kind of just maybe extending off of that, like get as many experiences outside of the classroom as you can. There's a lot of opportunity to help other folks do some research or, hop on the different opportunities. Like I said, yes, to go out, help tag monarch, but that was not part of a class. It's just everybody wants to help you at this stage learn different skills and explore what your interests are. So do as much outside of the classroom and then don't pigeonhole yourself into taking classes that are just within your major. Maybe if you think that there's some interest that you have, marine biology is so broad. You could really like take a class in engineering if you want and think that you might want to maybe merge some of those things or take a social studies class. I guess, yeah, maybe don't dive too deep too fast.
Thank you.
So Isabella Peterson asks, hi, what have you found to be the greatest difference between conducting research in an academic setting versus in a government agency?
Hi, Bella. Good to hear from you. Bella was a surge last summer and is now working in Alaska with USGS doing some pretty cool, I think this is like a fellowship that she applied for. So good testament to just taking the next step, one summer after the next. Bella, in terms of doing research as a fed, I technically am more of managing and understanding the research that's happening still in academia, just partnering with different teams that consist of academics and sometimes feds. But I would say that the folks that I'm working with, even within NOAA, there's a lot of collaboration that's happening. There's a lot of recognition that like what we're doing is more of a joint effort. And so the folks that I work with that are maybe doing the field work in the marshes are also coordinating with the folks that are doing the social science work, who are also coordinating with the folks who are helping to communicate the work. I think of a good way to say that, but I feel like it is just super collaborative and it's also very responsive to societal need because a lot of that work is being funded by taxpayer dollars and it's being, congressionally approved and supported. So yeah, a lot of it is very vibrant, results of tools and technical expertise that can be shared with communities. We can chat more about that later if you want.
Question about the snails. Do you think that parasites could be something that influences consumer resource interactions?
Knew you were gonna ask that one. So these snails actually do have a really cool parasite. As long as it's a traumatic parasite. I'm not as much of a parasite enthusiast, but I appreciate them. Somebody did some work in one marsh looking at snails and how grazing is influenced by parasite infection and found that if you're infected with a parasite, it actually increased your grazing rates. So really get another rabbit hole that you could throw yourself into is looking at snails and parasites.
Thank you. We have time for one or two more questions. Is that a hand?
No, this is probably kind of an outdated strategy, but I heard about this thing kind of similar with is it considered an invasive species, the snails, because their population is very part, would it be?
Like, is the Solidarity irrata considered an invasive species?
Yeah. Um,
Actually, I don't think so.
oh,
Not Littorina irrata, Littorina littorea is considered an invasive species here, the rocky intercoastal snail that you see, that periwinkle, but the Littorina irrata, I think it's considered a native species.
I mean, I know you said you didn't have an answer for like how what it was specifically causing the damage to the marshes, but I was just wondering if there was anything done to solve that problem or any attempts made to improve the conditions.
Great question. I think so. What my work ended up supporting is more of a nuanced look at how these snails aren't necessarily like the problematic top-down consumer in the system that's driving wide-scale marsh die-off. Like that does happen in like really punctuated circumstances when you've got drought and stressed out plants and a lot of hungry snails. That can happen. But for the most part, in a lot of these marshes, the ones that I worked at, the ambient snail densities are not really, they're not doing that. They're not having that effect that I could tell. And in fact, sometimes they're having positive facilitative effects on plants, and they're good things, helping recycle nutrients and dead plant material. But context is important, and as the environment changes, and maybe we have increased warming and drought and whatnot, that could change that relationship. So TBD.
Any last question for Dr. Atkins?
Also, when it comes to your career questions, I just really want to re-emphasize, talk to all the other people here. There's probably going to be at least one career panel while we're here. It's like I have some thoughts, but like it blows my mind when I hear what other people have to share about their path and suggestions to grad school and classes.
Can I ask you one last thing?
Yeah.
Where did the artwork in your slides come from, Rebecca?
Well, actually, I had backup slides. So beyond the research, she totally did not know this. I just had different things prepared if folks were interested in talking about different paths. Something you can also do, I did a lot in grad school, is I took scientific illustration classes. Also as an undergrad, I took a couple of art classes here and there. And that's just one thing that you can explore to help you with your science communication, or also just like, be like a full, well-rounded, happy person.
It's a really great example of you following the advice that you've given everybody here about following your passions. And so, yeah, please join me in giving Rebecca another round of applause. And we'll look forward to seeing everybody either online or in person for next week's talk. Thanks.